enables both identification and quantification of the peptides of interest at the same time. It has a good dynamic range because we operate the mass analyzers as filters, so we reduce the chemical noise. We can also have good signal to noise and high sensitivity, and we can multiplex, so we can measure not only one precursor but actually several peptides of interest within an SRM method. So what I was saying is that in SRM, so we do this sequentially. So first we measure one fragment ion, then the second one, then we change the precursor, and we fragment the first ion for this precursor, and so on and so forth. We have the cycle, and we would like to do it instead of sequential, whether we could do it in parallel. And this is what Bruno showed you on how to do parallel reaction monitoring, in which we actually instead, well, we measure all the fragment ions together in one scanning mode. Okay, so the thing is, this case in PRM, so we have the first mass analyzing in filtering mode and the second mass analyzer in scanning mode. And then we can actually align all these MS2 spectra that we acquire in each round and do the extraction of the peaks, as you have been practicing during the tutorials with SRM data and SWAT data, right? So we can actually interrogate how each of the fragments are eluting along the time, and then identify co-eluting fragments and therefore quantify and identify our peptides of interest. Similar to SRM, so here we also have advantages. So PRM allows us to both do identification and quantification of our peptides of interest, and in this case we also have good dynamic range, good signal to noise, and high sensitivity. But in contrast to SRM, in which this comes because of the filtering mode, here it comes because of the high resolution. And also you saw all these talks or all these slides from both Jared and Bruno on how the resolution actually changes the number of interferences and the limit of detection of your acquisition method. Okay, and then once we saw this, so both SRM and PRM, we move towards high throughput, right? And the question here is, well, in PRM or SRM, we are seeing parts of this so it's a targeted acquisition mode, so we are only identifying certain precursor ions, and the idea would it be possible to monitor everything all the time, right? And for example, so it has been mentioned here that strategies like MSC, where we have both mass analyzers, but they are not in filtering. So here, this is operated as a filter, but actually we are letting either pass everything, so the whole mass range, or specific mass ranges of a window of 20 m/z. And the second mass analyzer, we are similarly to what we are doing. So what we have are complex MS2 spectra formed by precursor from fragment ions from different precursors, right? So this is the MSC, and we also have the, for example, swath type of data, in which instead of isolating the whole mass range, we do it in windows, right? But the strategy is exactly the same. So we can align, as you have seen during the course, so align these MS2 spectra and actually then ask which are the fragments that are co-eluting in time, right? And in this way, identify the peaks. And we have seen, or it has been mentioned, especially in Jared's presentation, that there are several ways of extracting this data, right? So we have seen Skyline, but there is also OpenSwath and other like XDIA, and recently DIA Umpire, right? In this case, and this was also commented in one of the talks before, is that here we mine or we query the swath data, data-independent data, not in a targeted form, but actually we take all this data and ask, well, what is in there? Coming back to kind of similar approach of what we were doing in Shotgun, okay? And so many, or during the course and also in different proteomics forum, the question that arises here is data-independent acquisition a target mode, or is it a shotgun mode? Right? So if I'm targeting everything and I can actually extract whatever, then we go to our samples and say, well, what do I see here? And then we are back to our initial of the shotgun data, right? With different type of data, but actually I'm not so sure whether this is targeted or not targeted, right? And this is one point that I would like to discuss after these few slides, right? Again, right, so this data independent has some advantages like identification and quantification in this, right, at the same time from this data. So from the MS2, you can both quantify and identify the peptides of interest, improve dynamic range, high sensitivity, and multiplexing. So virtually, we can actually multiplex. So we are acquiring everything, so we could have data for all the peptides in our sample, right? And here, right, comes the question on what is target proteomics, right? Is it shotgun proteomics? Is shotgun proteomics or like top-end methods targeted proteomics? Is swath or data-independent acquisition, is it targeted proteomics? Okay. Before starting the discussion, I also wanted to maybe review the shotgun proteomics schema, right? And you know there that we first do an MS1 in which we operate the first mass analyzer as a screening mode, and then we select, for example, in this case, the 3 most intense precursor ions, and then we operate the mass spectrometer as a filtering mode, so we isolate our precursor of interest, in this case, the red one, and then we do a scanning, we fragment this peptide, we do a scanning mode in the MS2, and we get an MS2, right? So we have identification for the red, for the green, and for the blue. Actually, this data, I can also see it like in this way, right? So I have MS1 and a bunch of MS2, MS1 and a bunch of MS2. And I have in a bar, which indicates my cycle time in black, right? And what I can do conceptually is also remove all the MS2. I say, well, I don't care about them. And I align all the MS1, right? As I was doing for, or we have been doing for PRM or for swath data. And then I can do extracted ion chromatograms, that it has been done for several years with, for example, Progenesis or in Excalibur, that you say, well, is this mass in my sample, yes or no? And we can do this in the chromatographic dimension. And actually, we can also then get this type of maps where we have time or retention time, mass over charge. So each of these circles is a feature, a peptide. Colored means which is identified. In gray, it's not identified. I can align these maps and share data information. So I can also get virtually complete matrices of peptides identified in all my samples and quantified with MS1. Right? And this has been done for several years now. And question would be, again, is this targeted proteomics? Right? So is what we have been doing during the last 15 years, these shotgun top-end methods, are there targeted proteomics approaches or not? Okay? And now I would like to know your opinion. So,
well, I actually think it's semantics. It's targeted proteomics. I think you can split that maybe into targeted acquisition and targeted analysis. So I think regarding targeted acquisition, maybe it's more like, yes, DDA might be targeted acquisition because, you know, the first quadrupole is fixed. So you you target one precursor, except of the MS1 scan. Yeah, so I think it's more targeted proteomics. I think is not really correct. I think you would maybe have to split it in targeted acquisition and targeted analysis. Targeted acquisition is, as I said, DDA and MRM is targeted acquisition. Swath is not targeted acquisition because you have a wide quadrupole. Regarding analysis, DDA is not targeted. Swath and MRM are targeted because then you're really interested in which peptide. Yeah, you basically only analyze those peptides you're interested in. This is how I perceive the situation.
But for example, for swath analysis with this dia umpire, so now you can ask, so you have everything there, and you can ask, I want to know or extract everything, all the information that I have. So I extract all the peaks, I do a quality control FDR assessment of peak picking, and then there is no question anymore. So you get all the peptides that are, or for which you have enough information. So would this be targeted proteomics?
No, then I would also say this is untargeted, and it depends on how you analyze the data. This is what I think.
More opinions? Brandon.
So I'm going to introduce two new terms into the discussion. Actually, we've talked about them this week. One is peptide-centric analysis, and so that's what we're talking about in the McCoss lab a lot now these days. So there's so peptide-centric means that we're querying our data for peptides. So we start with a known set of peptides, and we ask the data, are those peptides there? And so statistically, that's a very different question than data-centric analysis, where we're at, and our classical shotgun has been data-centric. It's we go to our data and we ask, what's there? What do we see there? So progenesis, when it's finding features in the data and then matching them up to peptides, is data-centric. Swath, even if we're querying 100,000 peptides, if we're starting out by querying peptides, we're peptide-centric. And so traditionally, we think of this as being targeted, and this is being discovery. Although, well, I wouldn't even say discovery, but this is targeted. This is not targeted. But then the other axis on which we analyze, on which I think that we view targeted versus untargeted is what we've been talking about with so hypothesis test versus hypothesis generation. So do we go in with a hypothesis that we're actually testing, or are we asking the data to give us a new hypothesis? And so I would say that in this respect, swath experiments that start with 100,000 peptides and so on this axis we would say this is targeted, and this is really discovery. And so this is where I think the confusion lies. I don't know, if both of these are targeted, then, if we decide we're gonna call all of this targeted, then that gives us a certain set of things. Or we could be stricter and say, oh, sorry, if you don't have a hypothesis, you're not targeted. But certainly, I think we would all agree that this is not targeted, and this is not targeted. And that's my interpretation.
Oh, thanks. Yeah, so important thing as a follow-up with Brandon, so is that actually, for example, you can have swath here and also swath here. This depends on how you analyze the data. So saying that swath acquisition is targeted proteomics is not so clear for everyone.
You can put swath.
Also there?
Yeah,
well,
sorry, may I interrupt you?
Yeah.
Can we exchange your microphone?
Yeah, sure.
It seems that your headset is not really working.
Okay.
Or it's running out of battery or something. I'll just keep it. Okay.
Yes, so, for example, swath is everywhere. So, so is swath then, well, who would vote swath is target proteomics or not? Or Hmm?
What to call swath. So swath was actually coined by ETHZ, but and so when you are measuring DIA with the thermal instrument QX active, you are kind of also using these M/Z intervals. Then it's swath, and DIA could be MS to the E also. So is that kind of the difference, or is swath kind of an analog definition of DIA? Where do we have to draw the line then?
Maybe I can point something out about it. There's some confusion about this swath name. It wasn't actually Sykes that came with this name. It came from us. So it was actually Ludovic Gillet's wife, who Ludovic gave the talk on about M-Profit. And his wife is some kind of a geophysicist, and they do this swath satellite acquisition. So it actually came, and AB decided to go ahead and register a trademark, and then this has caused some confusion. But when we refer to swath, it's kind of as described in this 2012 paper from Ludovic, and you can do with that what you like. Brandon.
So I would say that swath, DIA, I just see them as synonyms. To me, DIA is just the, doesn't get me in any legal trouble with AB/SciX, so I just tend to use it. And that I think that's the spirit in which Jarrett and pretty much everybody outside this lab who doesn't feel like, hey, we came up with this name. What do you mean trademarking it? I think we just are having to use DIA as a synonym. So when you hear DIA, I actually frequently just write DIA/swath and hope that doesn't get me in trouble. But in terms of it's an acquisition method. It's an acquisition scheme. What you do with it then, I mean, you can do targeted analysis of DDA data. You could take DDA data and if you just go in and have a hypothesis and you test that hypothesis using the chromatograms in the MS1 scans, that's targeted to me. Similarly, with swath, certainly if you're going in and querying about specific peptides, that's targeted. If on the other hand, DIA umpire is again, it's just asking the data what, okay, we're gonna extract the data, we're gonna do a classical peptide spectrum matching thing, ask the data what's in this run. So we're going in from the other direction where we let the data tell us what the peptides are, which is extremely powerful, but that's the side that's not targeted. But that can be done on swath. So it's really about how you use the data and not how you acquire, what you acquire. I mean, there are certain acquisition methods where you tell the instrument up front, like PRM and SRM. Those are by definition targeted because you can't do them without targeting. And then the survey acquisition methods, both of them can be analyzed with targeted analysis or not.
So would anyone say, well, maybe this is better not to be recorded. Yeah, so would anyone say that, for example, at the MS1 level, this is data independent? So we are acquiring in a data independent mode at the MS1 level? Would you agree on this? Doing a similarity of what we are doing in swath, that we are acquiring all the mass range or windows at the MS2 level. So swath or other data independent acquisition modes at the MS2 level. So we acquire a portion of the M/Z range, and no matter what is going into the instrument. So we say this is data independent at the MS2 level. So here, if we do the analogy, we would say, well, no matter what it enters to the mass spec at the MS1 level, this would be also data independent. Well, this is just to get some discussion. So So would a traditionally top N method, could it be a data independent at the MS1 level?
What is a traditional top N method? I don't understand the traditional.
Oh, top 20, for example.
What does that mean?
This, well, so here we have a top 3, right? So I have data independent acquisition at the MS1 level, so no matter, and then I select the precursors to be fragmented based on the 20 most intense peaks in the MS1, so data dependent at the MS2 level. So for
What do you do with the data?
I don't understand the question now.
That's a survey acquisition scheme. That is an acquisition scheme. You can analyze it. So survey acquisition schemes are by definition not targeted. They're not, they could go either way. And
Yeah.
that's like, now, are you going to analyze it in a targeted way with a hypothesis that you're testing, or are you going to analyze it in a way where you're asking the data to tell you what's there? And then I would say I can answer your question once you tell me that.
Well, so here, what one can do is actually extract, right? So extract chromatographically the peaks at the MS1 level. So what I would call this is a data-independent acquisition mode at the MS1 level and data-dependent mode acquisition at the MS2 level. This is how I see, for example, this type of experiment, right? So it's not either one or the other. It depends on which level I see. So whether I see at the first mass analyzer or in the second mass analyzer, and so how I operate both mass analyzers depending on what is entering in the mass spectrometer or in the collision cell.
So just for me to summarize what the both of you said is that if we want to be always super clear about what we are doing and for what we use the technique, we would need to generate super long names where we specify what are we doing in the MS1 for acquisition, what in the MS2, and what do we do with the data afterwards in terms of analysis, right? Then we would always be really clear about our mass spec data, and that the titles of the papers will be endless.
Yeah. So I completely agree with what Tina has said, and I think this is one of the most problematic things in proteomics, right? So that we made names that just summarize part of the method, and then gets confused. So it generates confusion to the users or to the experts in proteomics, right? Until you actually go into the method and say, oh, now I understand how we operate the first mass analyzer and the second mass analyzer, or how we generate or the data and how we analyze it. So I completely agree that, let's say in an ideal qual, we would actually do this, so name or term all the methods and target or data analysis strategies completely. I think it's important to have both things. So these acronyms, right? So that facilitates communication, but in your heads, always think how I'm operating the mass spec and how I'm analyzing the data, right? And in this way, we can separate or say this same acquisition method in some cases can be a targeted proteomics approach, and in some other cases can be a non-targeted approach. Okay, and just to summarize and conclude this session, so let me also show, so this is not, so let's put it this, so this is not final or something written in fire, right? So, but this is something that helps me actually to name or to understand targeted proteomics, right? And I separate it in MS1 level and MS2 level. So this would be 1 one classification, so whether it's targeted at the MS1 level or whether it's targeted or untargeted at the MS2 level. And then whether I'm doing targeted acquisition or targeted data analysis as it was pointed before. And so to summarize what you have been seeing in the course, so I would say, well, for example, an old method that is targeted at the acquisition level with MS1 would be, for example, the SIM, so the selected ion monitoring. So I'm operating the first mass analyzer as a filter. I monitor just the mass of my precursor over time. Then similar thing would be targeted at the MS2 level with SRM or even PRM. PRM is a bit special because it combines both things, right? So it's targeted acquisition, but we also do targeted data analysis. And finally, so the MS1 targeted data analysis would, for example, if you look for specific peptides in a MS1 map, or, for example, if you look for specific targets at the MS, also the MS2 maps generated by data-independent acquisition modes. Okay, so we reached the end of the course. Any question or, so now there are the certificates, so. Okay, good.
When you have a target. Either you have a target.